ACROPOLI is a precision oncology study led by the SOLTI academic group in cooperation with 13 internationally renowned hospitals. This is a multi-center phase 2 clinical study led by Dr. Aleix Prat from the Hospital Clínic of Barcelona and Dr. Josep Tabernero from the Vall Institute of Oncology. ACROPOLI, SOLTI's first basket trial, aims to evaluate the ability of the molecular biomarker PD1 to predict among 30 different tumor types which patients will benefit from immunotherapy. What is PD1? The immune system can regulate the intensity of its response through different control mechanisms, such as the interaction of the cell surface receptor, PD1, with its ligand, PD-L1. This way, different populations of immune system cells are able to communicate and regulate their activity to prevent it from being excessive. On the other hand, tumor cells have developed sophisticated mechanisms to escape this immunological control, such as expressing themselves the PD-L1 ligand on their surface, and thus preventing them from being eliminated by the immune system. These observations have driven the development of inhibitors of the PD-1-PD-L1 axis with the aim of preventing tumor cells from using it to survive. Spartalizumab or tislelizumab are an example of them. They are humanized antibodies that block PD1 and that have demonstrated clinical efficacy in previous studies in patients with different types of solid tumors. The ACROPOLI trial aims to predict the benefit of treatment with spartalizumab or tislelizumab regardless of the type of cancer, and in a very innovative way by previously measuring the levels of PD1 mRNA in the patient's tumor samples, which represents a paradigm shift with respect to the current approaches and methods, which do not take into account the molecular characteristics of the tumor and its microenvironment. A total of 13 hospitals nationwide are identifying patients with one of the 30 types of advanced cancer that are included in the study and who are candidates to be treated with immunotherapy. Prior to their inclusion in the study, patients are pre-selected based on PD1 expression in the tumor. In the ACROPOLI study, patients are distributed into 3 cohorts. Cohort 1, patients with high expression of PD1. Cohort 2, patients with low expression of PD1 who both receive spartalizumab. And Cohort 3, patients with high expression of PD1 receiving tislelizumab. Currently, Cohorts 1 and 2 are no longer including patients, but Cohort 3 is actively recruiting. Patients participating in Cohort 3 of the study will be treated with tislelizumab intravenously every 4 weeks, and they will be monitored with the aim of evaluating the effectiveness of the treatment and the possible appearance of adverse effects. Additionally, other promising immunological biomarkers in the tumor and blood will be evaluated during the study. The results obtained in the ACROPOLI study could have great clinical value, covering the current unmet need to identify reliable and reproducible tumor biomarkers to predict who could benefit from immunotherapy, regardless of the type of tumor. Furthermore, the study would have a high impact on society, giving access to this drug for patients with certain types of rare tumors, where it is unknown if immunotherapy is effective, and, thanks to the study, we will know if spartalizumab or tislelizumab could be useful therapeutic options for them. If you want to be part of this project and obtain more information about the ACROPOLI study, you can contact us at our email